Hello and welcome everyone. In this podcast we're going to talk about inheritance patterns of the X chromosome. So let's write X chromosome inheritance. We've talked about this before when we talked about pedigrees. And just to remind you a few things, we know that if a male and female have kids, and if the male has it, since he's XY, we know that he only has the X chromosome that's mutated. And so we know that he will pass that on to all of his daughters. So all of his daughters will be carriers. And while his sons don't receive one of his X chromosomes, they will be unaffected. And in this scenario, we're assuming mom here is XX homozygous dominant. She doesn't have the trait. So we know dads give it to all of their daughters and none of their sons. Now what if mom has it? We know that mom will give it to, as a carrier, all of her daughters will be carriers, and all of her sons, we'll give her another son here, all of her sons will have it because they get their only X chromosome from mom. And we also said because of this, males are more likely to have X-linked diseases. So these rare X-linked recessive diseases are fully expressed in males when they only have one X chromosome mutated, because they only have one X chromosome. Females are less likely to get this because they have two X chromosomes. Now initially we didn't know this. This we learned over decades of research. And the first person to stumble upon this idea that the X chromosomes are inherited in a slightly different way, or the phenotypes associated with X chromosome inheritance were a little different than those of the autosome. So let's bring back our good friend Morgan. Morgan, remember, he was the first geneticist, he was the first person to have received a Nobel Prize for genetics. And in a very interesting story, though I never met him, of course, he helped me get into graduate school. That's a story for another day. But remember, him and his students, Sturtevant and Bridges, were working on fly mutants. He was really interested in how organisms change. And he had the idea that if you put a fruit fly in the dark where they weren't going to use their eyes, and they had many generations, that eventually it would no longer have an eye. Now interestingly, this might actually work, but you'd have to go through many more generations, years worth of generations probably, to generate that. Because at some point, if you're not selecting for the eye, you probably would lose it. But he didn't do it that long, and that's okay, because he stumbled on something else that was really interesting. Now this interesting result that he ended up obtaining was that he obtained this white-eyed mutant. He knew if he took this white-eyed mutant and crossed it with another white-eyed mutant, that they would all be white-eyed. There are rumors that Morgan kept these white-eyed mutants at very close proximity to him at all times, because he found them to be very interesting and he thought they might hold some interesting questions for the future. So to further analyze this white-eyed mutant, he started crossing it with red-eyed wild-type flies. And just like Mendel had done decades before Morgan came onto the scene, he started crossing white-eyed males with red-eyed females, and then white-eyed females with red-eyed males, assuming that he would get the same results, because he believed that this would be on an autosome and that the genes would assort independently. So let's first write out what we would see if it was an autosomal gene. And let's say he obtained the heterozygotes. So he took a true breeding white-eyed with a true breeding red-eyed, and he crossed them. And he would have obtained something like this. Let's use R for the gene. White might be a better choice, but sometimes it's hard to tell the difference between uppercase and lowercase w. So if this was autosomal, this is what you would get. You'd get one red eye, RR, you'd get one heterozygote, big R, little r, and another big R, little r, and then you'd get this homozygous recessive. So you would expect 3 red-eyed to 1 white-eyed. You'd expect this, 3-to-1 ratio of red to white eyes. And you should get that same result if this one were female and this one were male, or vice versa. However, that is not what he saw. He saw something different. Well, it turns out, as you can imagine, that this gene for red-eyed phenotype wasn't on an autosome. It turned out to be on a X chromosome. And as a result, we have slightly different predictions to make. And so what I want to do is to follow the first cross to completion here. So if he took, as the original P generation, a female with red eyes, so the gene was not mutated, and he crossed that with a male with white eyes, so it's mutated here, to get his F1 generation, what he would end up with, and we can do the little Punnett square over here, so we'll go X plus, X minus, and Y. What he would get is our progeny like this. All of the males would have red eyes, all the females would have red eyes. Alright, now, at this point, he would have looked at these results and thought it still could be on an autosome, because he's getting all red eyes. So he would take these F1 progeny, he would take the males, X plus Y, times the heterozygote here, of the females, and what he would get would be something like this. It's still a 3-to-1 ratio of red to white, which is what you'd expect for an autosomal mutation. But what's interesting and different is that the ones that had white eyes, they were all male. Never where it was a female found with the mutant characteristic of white eyes. So this was a first thought, first indication that the source of the mutation matters. If the mutation comes from mom, or from mom here, or from dad here, it matters. In this case, the mutation came from dad, and so in the F1 progeny, I had that listed. These are F1s, and these are F2s. The progeny are F1, we wouldn't see any difference. There is one, but phenotypically you wouldn't be able to tell. And it's not until you get to your F2 generations that you see that there is a difference. Only males have this trait. So now let's do the reciprocal cross, where we take the white-eyed female, because they both have a mutation on them, and the red-eyed male here, and we cross them. And let's do a Punnett square here. The females here, the males here, and we end up with something like this. Now in this case, even at the F1, we realize something interesting is going on here. Because what we find is we have a 1-to-1 ratio. Half the progeny have red eyes, half the progeny have white eyes. Now we might look at that and say maybe it's an autosomal dominant mutation. But what we see here is of the ones that are white-eyed, they're all male. And the ones that are red-eyed are all female. Anytime you have this gender bias in the results, you should be suspicious that what you're looking at is something that is X-linked. Or in some cases it could be Y-linked, but we're just talking about X-linked now. Now if we take these F1 progeny and we cross them together, what we get is again another 1-to-1 ratio, 50% red-eyed, 50% white-eyed. But of the white-eyed mutants, half of them are females and half of them are males. So again, by doing these reciprocal crosses, we get different results. And the numbers don't make sense for it to be an autosomal mutation. So anytime you do a cross and you start analyzing the results and you see a gender bias, as we see here in both the reciprocal crosses, that should be a clue that it's an X-linked trait. If you're presented with a problem and you follow all the data out, that's what I'm going to be looking for. Can you interpret the data to tell me if it's X-linked or if it's autosomal linked? Alright, that's all I have for this podcast. If you have any other questions, please let me know and I'll be happy to answer them. If not, I'll see you in class. Bye.